Hey everyone, welcome back to another episode of Behind the Science. I'm here with Paul and Monira in their recent paper in JACC, Loss of Y Chromosome and Major Cardiovascular Events in a Prospective Study of Older Men. It's a great paper and I'm happy to have them on. Maybe Paul and Monira, you guys could just start by explaining the paper and the main findings.
Sure, very happy to, Adith, and thanks for having us on today. So our paper describes a phenomenon called the loss of Y chromosome, which is the loss of the entire Y chromosome from circulating leukocytes or white blood cells. And this happens as men age. So it's actually quite a common phenomenon after the age of 70 in men. Between 20 to 40% of men at that age will start losing the whole Y chromosome from a significant proportion of their leukocytes, say from 10% all the way up to 50% of their circulating leukocytes. And this phenomenon was actually discovered by our colleague Lars Forsberg from Uppsala in Sweden about 10 or 15 years ago. And it's since been studied across a range of epidemiological settings and disease pathologies, and it's been shown to predispose older men to a range of different cancer types, blood cancers and solid tumors, as well as Alzheimer's disease, macular degeneration, and other types of cardiovascular diseases. So it's really emerged as this biomarker of aging and a risk factor for various types of diseases. However, in the setting of cardiovascular disease, most of the prior studies have been performed on loss of Y chromosome in men with existing cardiovascular disease or chronic kidney disease cohorts. And what we really wanted to do was see whether detecting the loss of Y chromosome in initially healthy older men would predict future risk of cardiovascular disease event prospectively. And that's what our paper does, and I think that's the new contribution that it makes to the field. And we were able to do this through analysis of the ASPRE clinical trial cohort, which was really the perfect cohort to answer this question. So the ASPRE trial, as some of the viewers may know already, was a large randomized clinical trial of low-dose aspirin in individuals over the age of 65 in Australia and the US. And it was a primary prevention trial, so looking at whether low-dose aspirin prolongs disability-free survival in otherwise healthy older people. And the trial recruited 19,000 participants, so it's a very large trial and community-based. And the key point is that all the participants had no history of cardiovascular disease of any type at enrollment. Yet majority of them were over the age of 70. The average age was about 74 at enrollment. So we had this very unique ascertainment of healthy older people without any cardiovascular disease. And we were able to get about 75% of them enrolled in the biobank program to give a blood sample at baseline. And that was key because it allowed us to do all these studies, including genetic studies, but also measuring of biomarkers at baseline, the time which they were healthy, to look at what measures in those samples predict future risk of disease, specifically cardiovascular disease, where we had very good confirmation of the absence of cardiovascular disease at baseline, which was done by general practitioners who assessed their health status in order to enroll them and randomize them into the trial. So that, as I said, that created a perfect setting for us to measure loss of Y. And we had already genotyping data on the whole cohort available, which we can use to measure the proportion of cells that lose the loss of Y chromosome. And so we got into a collaboration with Lars and others from Uppsala to kind of put these two pieces together. And that's what we did, and we report the results in JACC, and I'll hand over to Monira to go over some of the main findings and results.
Thank you, Paul and Adith. So in this paper, we have looked at, as Paul mentioned, in a healthy bunch of people who were free from cardiovascular disease and overt hypertension, that with a loss of Y at baseline predicts MACE and its components of MACE, and we found that there is an association between MACE. And when we looked at the components, we saw that the loss of Y is associated mainly with myocardial infarction, not with ischemic stroke. This was a very intriguing finding to us because we wanted to actually check that in previous studies there were all these unhealthy people, but for our study these were healthy people. And how about their other cardiovascular risk factors? So we went through step-by-step adjustment. We know that age and smoking, they are the predictors of loss of Y. So at the first model, we have adjusted for age and smoking. And then we started to include all those cardiovascular risk factors like the systolic blood pressure, the blood cholesterol, and then their medications, allocation to aspirin, testosterone, frailty status, and estimated glomerular filtration rate. And we figured out that even after adjustment, these association persist. Also, we wanted to check, which have been checked in other papers, that the association in the linear association. So loss of Y has been looked at linearly, and then we tried to look at the gradient effect as well. So our data was very big enough and we could do this, and we figured out that we have created these categories based on the empirical evidence, and you will see that there are some negative values which are artifact of the machine. And we have figured out that when people do have less than 5% of the cells with loss of Y chromosome as one category, then 5 to 22% of the cell as moderate loss of Y category, and then we have thought that the rest who do have more than 23% cells without loss of Y as the substantial category, and figured out that there is a gradient effect, and the effect was very highest in the substantial loss of Y chromosome. So this kinds of signals us that yes, there is a dose response, and this loss of Y chromosome, if you do have some high level, you are at higher risk.
Yeah, that's a great point. Go ahead, Paul. Yeah.
I would just add to that. Yeah, so we think the results are pretty robust based on all those adjustments. But of course, the peer reviewers wanted even more than that. So they wanted to know whether there was an interaction with clonal of indeterminate potential, or CHIP, or whether so we then adjusted for CHIP and excluded all participants with CHIP and showed that the association with loss of Y and MI was still robust to that. And they also wanted external validation in an independent cohort. So we performed the external validation in the UK Biobank and found the same association in the UK Biobank for MI as well, which you'll find in the paper.
So I think the interesting point that you guys show is that there's this signal with non-fatal MI, but not necessarily with MACE or with ischemic stroke. And then Monira was sort of describing this 10th decile proportion of patients, which is 23%, plus, loss of Y. And in that cohort, right, compared to the first through sixth decile patients, the hazard ratio fully adjusted is 1.68, the 95% confidence interval 1.16 to 2.45 in the ASPRE trial. And so, but for ischemic stroke and MACE, that 95% confidence interval overlaps 1. So I just love to hear you guys speculate a little bit in terms of why you think that is, and maybe biologically, or maybe it's a signal thing or sample size, but that's sort of an interesting finding of the study.
Well, we thought the same way the question is framed when we saw the findings. First of all, we tried to check that what is actually happening. So it could be a number issue, that's true. But again, if we look at the biology, the stroke pathophysiology and the MI pathophysiology are very different. So MI, we know that it is from the rupture of plaques. And then when the atherosclerotic plaque ruptures, there is the inflammation. So all these things are with MI. On the other hand, when we think about the stroke, it is emboli from different big arteries. So it will go with the atrial fibrillation or things like that. And also, we also tried to look at that only the mechanisms that we can see. Is that the only plausible cause, or other studies has looked at the similar sort of difference with exposure and the outcome? So we figured out, even in our ASPRE, that Lp(a) that goes with MI but not stroke. Similarly, in the UK Biobank, the LDL that goes with MI, not with stroke. And the other interesting thing, and which is one close to my heart, and I will do some more analysis on that, that is the CETP gene. With the CETP gene, we figured out that this is with from other cohorts on MI, but there is no effect on stroke. So I guess that there are the plausibility that these are two different mechanistic process. But again, yes, we need to interpret this result with caution, because in our stroke group there were fewer events.
Maybe I could just have you guys double click on the external validation with the UK Biobank. Paul mentioned that in as well earlier, but the signal size or the signal effect decreases, a little bit. And so I just wonder if that's a function of two things. One of them being that you guys are, sort of looked at 40 to 70-year-olds in UK Biobank, whereas in your guys' ASPRE study, it was 65 plus if you're a minority, or 70 plus. So if it's maybe just the age difference. And then second question is, what enrollment criteria sort of, virtually did you guys use for the UK Biobank, and how well did that recapitulate the enrollment criteria for ASPRE?
Yeah, very good question.
Very good question.
So I agree that the main difference is the age at enrollment. So in the UK Biobank, there's a lot of individuals between 40 and 60. The average age enrollment, I think, was 57. And loss of Y really starts to happen, 65 plus, or even 70 plus. And then the actual enrollment criteria was different from the UK Biobank. Whereas ASPRE, you had a clinical assessment of the lack of cardiovascular disease at enrollment, which didn't happen in the same way in the UK Biobank. And then the ascertainment of the events themselves were different. So UK Biobank being primarily based on ICD-10 codes, ASPRE being a clinically adjudicated clinical trial, standard. But also, as you raised, there was the need to kind of censor at baseline the UK Biobank data set for individuals with prior cardiovascular disease, which we did do. So we removed any we tried to, do you remember the exact criteria
Yes.
we used to
So
remove?
we have used the ICD-10 codes again there. So with ICD-10 codes, there are the code for cardiovascular disease, but we have just excluded them as a bunch, not as of ASPRE. And also, as you know, that the ICD-10 codes are good, but not as robust as adjudicated outcomes. So yes, there should be a little bit of misclassification, but the signal is there. And also, as Paul mentioned, that the UK Biobank participants were younger, so the loss of Y chromosome prevalence was not as high as ASPRE over there.
There's also a healthy selection bias in the UK Biobank, which is widely acknowledged. They had to send out, I believe, 9 million invitations to enroll 500,000 individuals in the UK Biobank. So there is underlying ascertainment, potential biases and reasons why you might see different effects in any one cohort versus the other. But we were very enthused to see that we were able to replicate the continuous variable result of loss of Y and MI and MACE in the UK Biobank, even though the effect was smaller. And also the effect was not significant for stroke in the UK Biobank, as it wasn't in ASPRE. So they was quite consistent in what we found.
And also, Paul, to add up here, I think that what we learned from here, that actually we can't use one or the other set of criteria for categorical variable yet for the population. So for example, in ASPRE, we could do like more than 23% of participants loss of Y in the substantial group. We wanted to apply that criteria in UK Biobank, but we hardly found any people in that group, let alone finding that association. And that gave us the initiation that, well, as we always are doing, that the continuous one should be our primary focus and the primary result. But then again, to find out the risk gradient, we can look at these different cutoff points as well.
But yeah, we're looking forward to seeing other cohorts and studies report on this and see how robust it will be across different populations in the kind of primary prevention setting.
Yeah, absolutely. And I think Paul mentioned this earlier with CHIP, right? The sort of, association with CHIP, whether or not it exists. And you guys did two things. One, you adjusted for CHIP, as you mentioned. And, it was the fully adjusted hazard ratio in, without adjusting for CHIP was 1.68 for a non-fatal MI. And then you adjust for CHIP and it goes down to 1.56, broadly consistent, just a little bit lower. And then you guys do the thing where you exclude CHIP, right? You throw out anyone with a variant allele fraction over 2%, and then your signal for MI is 1.9, right? And so I, would just love to kind of hear you guys' thoughts on, there's this population potentially of people without CHIP, right? But they have a high loss of Y, maybe 20%, maybe 30%. And that sort of suggests to me that that's like a uniquely high risk population. And maybe you guys just have thoughts on that and sort of maybe mechanistically on why that could exist or even clinically looking forward.
Yeah, so we're working on CHIP as well, and our original hypothesis was that if men who have loss of Y and CHIP will have a double whammy, and also do the two things occur together? Are they two versions of the same thing, two sides of the same coin, of like generalized genomic instability during aging? And we don't think that's the case. We actually think the opposite is true, that, and our data suggests this, and this is in a separate paper, but there's other studies that have actually already reported this, that if you have loss of Y, you're less likely to have CHIP than a man without loss of Y. And if you have CHIP, you're less likely to have loss of Y than a man without CHIP. So they're inversely correlated. So that suggests that they're independent or even competing processes that lead to disease in separate ways. However, more work's needed, I think, to fully understand, like, is this happening in different subsets of cells, like different blood cell types? And is that important? Is it specific to certain types of CHIP in one gene or another? But we think that the result that we found between loss of Y and MI risk is independent of CHIP and specific to loss of Y. And I think, the sensitivity analyses that we did support that.
So my sort of next question is a little bit more methodological, but you guys assess loss of Y through a microarray, and then you guys compare it to whole genome sequencing, and it's broadly consistent, right? The correlation coefficient is like 0.98, right? But there's this phenomenon that I see at the lower end, right? Like, for anyone with a loss of Y estimated below 20%, there's sort of a wide range. So someone with a whole genome sequencing of 0%, right, could have anywhere, looks like from 0 to like 15, right? So to me, when I look at that, one, I have a question methodologically: which one's the gold standard? Which one should we use? Second, maybe that's the reason that the deciles 7 through 9 compared to 1 to 6, because of this variability, we don't necessarily see a hazard ratio that's sort of, more towards the 10th decile versus the first, right? Sort of an underestimation of maybe the true effect. So those are kind of two questions hand in hand related to the methods of kind of assessing loss of Y that it would be great to hear your comment on.
Sure. Yeah, so the first thing is important to say is that loss of Y is a spectrum or a gradient effect. So there can be 1%, 2%, 10%, 50% of cells losing their Y chromosome. And then the way that we measure it, the most commonly used way has been microarrays. So I would probably say that is the gold standard. Not that it works perfectly, but more cohorts have that available. And the way that it works is you take the intensity signal on the SNP microarray, averaged across all the non-sex chromosomes that should always be in roughly the same copy number, and then compare it against an average intensity of all the probes on the Y chromosome, which will reduce relatively in men that get more Y chromosome loss. So your your Y signal versus your autosome signal is your ratio that we use to estimate loss of Y. And then you can do another estimation of the percentage of cells that might correspond to try and quantitate the percentage cell loss. And that's an estimation, right? That's not an actual measure. You can use flow cytometry and single cell sequencing and other methods now to get an actual cell by cell measure. But in a large cohort epidemiological study, that's not really feasible. So what happens is you get this estimate based on the signal ratio, and you can actually end up with positive values where it looks like you have more Y chromosomes than autosomes, which is just noise. So all of the stuff at the low end using the microarray, there's a signal to noise threshold there. And so we think a large proportion of men are just in that, noise group, and they have essentially no or negligible amount of Y chromosome loss. And then you get into the area where the signal separates from the noise, but it's still, again, not perfect. It's still an estimate. And that bleeds into this moderate group where we think that there's probably 5% of cells to 20% of cells losing their Y chromosome. And a lot of that may be non-pathological, or it may take another 10 years for that to increase to a level of signal that might represent a significant risk factor. But then you've got the men at the end of the spectrum, which we think have, over 23% of cells that have lost their Y chromosome, which is definitely signal and not noise, and really what we would describe as a kind of pathological risk factor group. And that's where we see the associations in. So it's definitely not a binary event, and where you put the cutoffs along that gradient is very important and will vary by cohort to cohort, especially by age, because it's a strongly age-related phenomenon. So because we had an older group of men in our study, we were able to do that with great granularity to get that group of substantially affected men. And then with the whole genome sequencing based measure, we had whole genome sequencing available in a subgroup of about 1,500 men, I think it was, or 1,200 men. And so there you've got sequence data on all the chromosomes, and you can look at the sequencing depth of the Y chromosome versus all of the autosomes across the whole cohort. And then you can see like drops in sequencing depth on the Y chromosome, which correspond to you can then estimate Y chromosome loss through sequencing depth data, which is also not perfect because you can get biases in the way DNA sequencing works based on the sequence context and other factors. But we're enthused to see that when we correlate those two completely independent ways of measuring the same thing, it's very highly correlated at 0.98. But maybe the array struggles with the low end a bit more than the sequencing does.
Yeah, that makes sense. And so I guess my final question to you is if you guys wanted to offer any mechanistic, speculations as to, this is a rather fascinating thing, right? That our white blood cells sort of lose a Y chromosome, if you're born with a Y chromosome, why that could be happening, and then finally what this means for sort of risk prediction in the cardiovascular realm going forward, and maybe how this could be used clinically, right?
Thank you. So on the mechanistic bit, first of all, Sano et al. 2 years ago showed that in animal models, loss of Y causes fibrosis. And in different muscles, including the heart muscle, especially heart muscle was mostly affected. And we thought that what are the mechanisms of heart fibrosis? So one of the major thing is the post-remodeling after MI. So we thought that it could be one of the mechanism there. Apart from that, there is the pro-fibrotic transforming factor B, which signals the Loy macrophage. Also, on the Y chromosome loci, there are some genes that affects the high blood pressure and also high cholesterol. And these are all like established risk factors of myocardial infarction. So we think that these are in the mechanistic pathway, but the real picture yet to resolve, because we don't know yet, but these are what we speculate. And the clinical utility you suggested, I think we are still far away from the clinical utility because we can't order loss of Y test in our laboratory yet. But what I feel like at this moment will be important that to include this loss of Y into the present cardiovascular risk scores, for example, the score OP, and check that how much prediction it improves. So if the prediction improvement is very high, at that time the test can be made available, or there will be other researchers who will be thinking in that way, that how this can be made available or things. So still for clinical application, it is coming, or it is coming ahead. But at this moment, I think it will be really good to look at in risk prediction how loss of Y actually improves or impact the risk prediction.
The other area is like therapeutics. Could you target loss of Y or try and prevent it? And our good friend and collaborator Lars in Uppsala in Sweden is looking at all these questions and the mechanisms. And I guess what fascinates me is whether it's causative or not, and how far back in the process is this event happening, and does it drive disease, or is it a kind of byproduct of disease? I think that's a very important question that still requires a little bit more work. But yeah, it's a fascinating field. I would still describe it as an emerging risk factor, and these age-related genomic changes in the blood, I think, are becoming increasingly important. And I just wanted to also give a shout out to our mentor, John McNeil, who was the PI of Esprit, who established the study, established the biobank, and has a great fascination in genomics and these age-related genomic events.
Awesome. Thank you guys. Wonderful paper, and excited to see what you guys do next and sort of follow the loss of Y story moving forward. Thanks for your time, guys.
Pleasure.
Thank you for having us.